let me go on now just to very briefly introduce tonight's speakers, James Lindholm. James has a BA from California Polytechnic, a master's degree in environmental science from Boston University, a PhD in ocean conservation program at BU. He's a fish ecologist and a conservation biologist. He has participated in 24 field projects, half of them as chief scientist at sites in the Atlantic and the Caribbean, the Pacific, and the Indian Ocean. He currently is science coordinator for the sanctuary and research scientist at the National Undersea Research Center at the University of Connecticut. And a personal note, I might add that I owe a special thanks to James for sharing air in his scuba tank while I was sitting on a decompression line with very little air left in my apparatus last November on a saturation dive trip with the underwater habitat called Aquarius. Our trainers for the mission had threatened dire consequences if we got down below 500 psi on our scuba tanks, and James allowed me to avoid their wrath. Clearly, James has a keen sense for how to use technology wisely. Please welcome.
Thanks, Ken. And thanks to the aquarium for having me tonight, and thanks everybody for coming. What I wanted to do tonight, obviously, is talk about some of the technologies we use in the offshore environment. As Ken mentioned, I'm a fish ecologist, so most of the stuff I talk about this evening will be related to studies of fish and fish habitat on the seafloor. There are a whole suite of technologies for pursuing other taxa that I probably won't dwell on much. Also, because this is focused on the sanctuary, most of the technologies that I'm going to talk about tonight we've used in the sanctuary, are using now, or will be using in the future. Some of the examples, like Ken just mentioned, the Aquarius habitat down the Keys. I'm going to throw in some tidbits from Aquarius, just because Aquarius is kind of a cool habitat opportunity. Because this is the first of the series dealing with the sanctuary, I wanted to just give a quick overview on the sanctuary itself. Go ahead, Ken. But first, I need to get out there with the acknowledgements. Everything that I talk about tonight, the technologies that we use, the projects in which they're used, have come in collaboration with these agencies, primarily the NERC, the National Undersea Research Center at the University of Connecticut. That's down in Groton, Avery Point area. The U.S. Geological Survey down in Woods Hole on the Hui campus. The National Marine Sanctuary Program in its entirety, our parent organization, NOAA, and for Aquarius Undersea Habitat, the Undersea Research Center at the University of North Carolina at Wilmington. Okay. So before Ken mentioned Stellwagen Bank, how many people in the room actually, let's see a show of hands, how many people knew about the sanctuary? Well, that is impressive. That's very good. We should turn the camera out to see the raised hands. That's impressive. I didn't anticipate that, and so we'll cruise through this intro very quickly. But just to put it in a national context, the sanctuary is one of 13 sites, extending from Stellwagen up here in the north all the way to American Samoa in the South Pacific. The sites are designated for a variety of reasons. The first site, which was the Monitor, was designated as a submerged cultural resources site. The most recent site up in Thunder Bay is also designated for submerged cultural resources. Most of the sites have some biological focus. The humpback whale is the only one that's focused entirely on a particular species. The remaining sites, including the next proposed site, which was designated by President Clinton as an ecological reserve at the end of his term, deal with the whole suite of biological taxa that occur there, fish, invertebrates, marine mammals, as well as cultural resources, and that's where Stellwagen falls out. Okay. So just to give a point of references for those few hands that didn't shoot up tonight, this is an exaggerated topographic map showing Stellwagen Bank proper that gives the sanctuary its name. This is Tilley's Bank, and this is Jeffrey's Ledge. So here you see the boundary of the sanctuary is Gloucester. We're out here in Boston. Here's P-Town. So the sanctuary is about 842 square nautical miles, which is roughly the equivalent of an area to the state of Rhode Island, for what it's worth. It was designated 10 years ago. This year is its 10-year anniversary. A lot of things have been going on. And this presentation is going to be relatively general, covering a number of different projects. Some of the forthcoming series will probably focus in on some of the particulars. Okay. And just to finally clarify, the goal of the sanctuary thus far is really to preserve, protect, and enhance biological diversity, ecological integrity, and the cultural legacy of the sanctuary, with cultural legacy including all the human activities go on out there. I mean, most of you are probably aware there's a great deal of human uses out there, from whale watching to commercial fishing, recreational fishing, private boating, all of these things. The shipping channel runs right through the sanctuary. There's a lot of human uses, a lot of interesting biological activity, and the sanctuary's goal is to reconcile all those ways such that the human activity can continue in a way that protects and conserves the animals found there. Okay. So to the meat of what I wanted to talk about tonight, I probably, like a lot of people, my experience with the marine environment was constrained to shore for the most part until I got into graduate school. And I realized then that the opportunity to work offshore is not one that comes to a lot of people. And one of the reasons why I wanted to give this talk tonight is to highlight that fact that I've, over the past year, I've interacted with the public a great deal. And I've also sat at a number of scientific panels where my fellow scientists around the table had very little to no experience offshore, no picture in their mind what the seafloor looked like. And that was stunning to me. And I think it's our obligation across the board to bring the marine environment home. So when you think about the challenges to marine scientists that probably aren't faced by our terrestrial colleagues, the first relates to the traditional mode of data collection. And this has been in the news recently. Some people are probably aware there was a bit of a hiccup with the National Marine Fisheries Service data collection approach. Much of the work that has been done traditionally with respect to fish populations and everything that lived underwater was conducted through trawl surveys. And we still conduct it. And I in no means intend to malign this form of data collection. It has its limitations. You drop a net over the side, drag it along the bottom, and count and index, measure, weigh, do everything you can to the things that you pull back up. But these are things that are usually dead by the time you pull them back up. There are fish, there are chunks of coral, bits of sponge, anemones and things. You're able to inventory these things, and we've learned an enormous amount by virtue of these trawl surveys, but there are inherent limitations. To borrow an analogy from a couple colleagues, Peter Oster and Elliot Norris, I analogize this. Please allow my graphic. I worked some time on this. Thank you. I thought the clouds were very nicely done. The analog for terrestrial data collection to these trawl surveys, were you to imagine yourself sampling a forest in the fog from a helicopter. Now you drop your net down, you drag it to the forest, you're going to bring up a lot of things in that net. There's going to be twigs, there's going to be squirrels, you might find a flower and a hummingbird squished at the back end of the net, but you wouldn't understand the fundamental connection between the hummingbird and the flower, which is that, and that ecological knowledge is critical for the conservation of both the flower and the hummingbird. So that's one of the challenges we faced right off with respect to the basic data collection. There's a secondary issue, though, and that one relates to, I've alluded to already, relates to the fact that there aren't a great deal of, the scientific community has no idea what it looks like offshore, and the same is true with your average citizen in the United States. Next slide. If I say to this assembled group, Yosemite National Park or Yellowstone National Park, you probably very quickly can conjure up an image of Wawona Half Dome or one of the geysers in Yellowstone. And you probably have a reasonable sense of the charismatic megafauna that occur there. If I say the marine environment, if I say Stellwagen Bank or the Mid-Atlantic Rift, you probably think of it, have a vision that's something like this. It's all blue from above, regardless of what's happening underwater. And with respect to the charismatic megafauna, you probably recognize it in this form. I do too. I'm just saying, I'm not again maligning eating of seafood. I'm just suggesting that in doing work offshore and in trying to bring that information back to managers in a way that it can be meaningfully incorporated into management, in terms of you the National Marine Sanctuary is in fact a national marine sanctuary. Sure, it's off the coast of Massachusetts, but these are held in the nation's interest, in the nation's trust. As we often say, the little old lady in Kansas has every bit as invested in Stellwagen Bank as somebody who's fished there for the last 120 years. So we have to overcome, through technology, this disconnect between what's happening underwater and how it looks from the surface. So I'm just now going to kind of roll off through some of the projects we've been involved in and iteratively move through some of the technologies that we use, and we'll see where we get. The first thing, of course, which is a major limiting factor working offshore, is that you need a research platform. You can't just go down like when I was in graduate school. A lot of my grad student roommates working in the intertidal, and they were able to go down to their sample sites every day, virtually all year. If it was snowing, if it was raining, it didn't matter. They were walking down to the tide pools, sampling the green crabs. Offshore, you need a research platform to get out there to do that. There are a number of opportunities available to us. One subset of these would be private or university research vessels. This is the University of Connecticut's new boat, the RV Connecticut. It's been in service for 2 years. 76 feet long. Basically fits the standard paradigm of a research vessel. It has dry lab space. It holds about 6 crew and 7 scientists at maximum. Can stay out for 14 days at a time. Has a reasonably large fantail space. An A-frame crane here for deploying gear. There's wet lab space. There's dry lab space. It's a reasonably stable platform for spending long periods of time offshore. Another option, particularly to us within the NOAA fold, is the NOAA Fleet. NOAA, National Oceanic and Atmospheric Administration, the people that bring you the weather service, also have a NOAA Corps, which is actually the 5th branch of the military per se. I guess they're not really military, but it is a 5th corps in addition to the Army, Navy, Air Force, and Marines. They have an entire fleet that are operated by the NOAA Corps. The NOAA Corps are the officers. The crew are often scientists or fishermen, depending on which boat you're talking about. This is the Farrell. The Farrell is 138 feet, and it's going to be decommissioned this fall. This boat, I personally have spent a great deal of time on this boat, many fun days being tossed around. The Farrell, for instance, all of these things have unique characteristics. The Farrell was designed for work in southern waters. In river deltas, etc. It only draws 7 feet. So for people who are used to going to sea, you could imagine being out at sea on George's Bank or in Stellwagen when the seas are 18 feet and you're in a boat that only draws 7, and your bunk is in this level up here. You get tossed around quite a bit. But on the other hand, it's got a huge fantail. It has an articulating crane, 2 A-frame cranes, a great deal of lab space. Both wet and dry lab space. It's an excellent platform for conducting a great number of research projects. And we come to the other end of the spectrum. This is the Sanctuary's boat, the Hawk, 30 feet long. There's nothing, you haven't lived until you spent a few days 20 miles offshore in a 30-foot long vessel. That's very exciting. Again, we have a small davit. This is our dive ladder. We do a lot of dive operations off this, and we also do a lot of the acoustic tagging work, which I'm going to describe. So with respect to vessels, you see we're working in anything from relatively small boats, 30 feet, to boats that are 150 feet or greater. Okay. Another option, and this is something that this is the notion of a saturation diving in an underwater habitat. This is Helgeland, which was located in 1975 on Pigeon Hill, which is up here just right outside the northern boundary of the sanctuary. Helgeland is a habitat designed in West Germany. It was operated by Poland at the time, and it was deployed here in 1975 for work on herring spawning. There's a great deal of herring spawning that occurs here on Jeffreys Bank. These habitats provide an outlet for what's called saturation diving. How many people in the audience have been diving before? Okay, a smaller number of hands. Everybody's probably aware that there are limitations to how deep you can dive for particular periods of time, and that's a function of a number of things, but primarily of the amount of nitrogen that accumulates in your bloodstream. Your body doesn't metabolize nitrogen. With a habitat like this, Once the human has been underwater for 12 hours, you're technically saturated. Your bloodstream can assume no more nitrogen. But as long as you stay at depth, you'll be fine. Coming to the surface would present a number of problems, but as long as you stay at depth, it provides unbelievable amounts of dive time. Helgeland, this was established in the winter of 1975. These guys were diving in wetsuits in the Gulf of Maine in the middle of winter out of a habitat. It was pitch black down there. These guys, it was, as we often say, real men. Next slide. And contrary to that is one of the descendants of Helgeland. This is down in the Keys. This is the Aquarius undersea habitat. There have been a series of habitats from Helgeland forward and a couple prior to that. Aquarius is right now the only operating habitat for research in the world. It's at 60 feet off, about 4 miles off of Key Largo, and provides 10-day missions that Ken and I have both been on recently that allow you up to 8 hours a day of dive time. So you're talking about a situation where if you were to dive from the surface, you would probably get 2 50-minute dives at max. When you dive out of the habitat, coming in and out, next slide, coming in and out of this moon pool, you just swim in and out, you have 8 to 9 hours of dive time, which dramatically increases the amount of science you can get conducted. It's ridiculous. In addition to just the data you collect, there's all this the opportunity for qualitative observations. I mean, for those of you who dive, realize, particularly when you're doing research diving, you're in the water, you're swimming right to your site, you're collecting your data because you have limited bottom time, and then you're swimming right back. No opportunities to kind of hang out and look around and check stuff out. Well, when you're swimming back and forth from Aquarius, this is what we call a way station. It's 1,000 feet. It's strung out along a line 1,000 feet away from the habitat. You can fill up. You can stand up in here, have a power bar, get a drink of water, fill up your air tanks. They have designed whips that come out from right here. You don't have to take your tanks off to fill, which is nice because they weigh 100 pounds. You're wearing twin 100 cubic inch tanks, I guess. But swimming back from this thing at the end of your dive, you're swimming 1,000 feet, which is much longer than you'd normally swim on a regular dive. You're able to see a whole number of things, interactions between animals. When you're down in the water for 10 days, swimming past the same fish, the same reef structures, you start to recognize behavioral attributes of these animals that you don't see when you're regularly doing work. We were down. I've been very fortunate enough to do 3 missions to the Aquarius in the past year, and I don't expect to be doing many this year. But nonetheless, there was one instance in which a storm passed through, and we were stuck in the habitat for an entire day. That was not fun. The habitat is, as you can see, this is the main area. We have 1 table to seat, and it holds 6 people. You can't sit all 6 at the table at the same time. Back here are the bunks. Everybody sleeps back there. You have a microwave, an instant hot water faucet for freeze-dried hiker's food, and there is a functioning toilet, but we're not going to talk about that for reasons that I'm not going to talk about. But the point being that after we were stuck in the habitat for a day, we came out after that day. The visibility was dropped. This is the Florida Keys. You'd think it's 100-foot visibility. Well, it was more like 25, something much more similar to a good day up here. But what we saw on this particular dive was remarkable. Hogfish, which are relatively solitary creatures, swimming in schools of 500 plus. Blue tangs, which you sometimes see in pairs, literally herds of blue tangs swarming over the reef. Gray angelfish, French angelfish, huge aggregation, not aggregations, these are schools, oriented similarly, swimming along the bottom, doing something that we didn't understand, but something that we probably would have never had a chance to see. If we wouldn't have probably been out there had we not been living underwater. So in addition to the fact that I probably did 4 times the amount of work that I would have done from the surface living in the habitat, all these qualitative things you get to see that you don't normally get a chance to see. Okay. All right, so we're out there on our platforms. We're diving from boats, we're diving from habitats, we're diving we're in the water. Underlying just about everything that we do out there is most of the stuff I do is our seafloor maps. Again, my terrestrial colleagues, and I don't want to appear like a whiner, but my terrestrial colleagues have, a series of maps that have been developed 100 years ago, have been fine-tuned over time to go pinpoint where they want to go. They can return to the same point in the same forest year after year. In the marine environment, that hasn't necessarily been the case. We haven't had that opportunity very much, but that's starting to change. The way that's changing is not a new technology. It's called multibeam mapping. It's a sonar. It's a multi-beam sonar. And here, this work, this red area here, which encompasses the sanctuary map's not on here, but it's roughly like this. This work was started in 1994 and completed, the mapping part was completed in 96. It was done in conjunction with the sanctuary, the USGS, and the National Undersea Research Program. The boat that was used was a twin-hulled swath vessel called the Creed from Canada. It's a Canadian Hydrographic Survey. They had a hull-mounted, meaning there was a system, a Simrad EM1000 multi-beam system mounted to the hull, and they literally mapped the entirety of this area going back and forth multiple transects over 2 years. You can see this, you see some striations in the data down here, and that's actually an artifact of the way the transects were conducted. Some of these striations up here that are harder to see at this scale, which is 1:60,000, you can actually see iceberg scour from 11,000 years ago. You can't see them when you're on the bottom because they're relatively gradual features. But at this scale they show up. It's really pretty remarkable. So with using this type of a system, which for those of you who aren't familiar with multibeam, it's a system that propagates sound down to the bottom. Multi-beam projects multiple beams, and in comparison to side-scan sonar, which I'm going to talk about in a minute, it gives precise georeference depths for every return signal. So by measuring the distance or the length of time it takes for a single pulse to hit the bottom and come back, it calculates the depth, and it gives a precise georeference point. It's my understanding that the resolution of this map is conservatively estimated at about 10 centimeter vertical resolution. That means that you can pick up something that's 10 centimeters in height and probably 10 meters horizontal resolution. So you can pick up a number of features. You can't see it necessarily at this scale, but they're there. Compare this to some of the mapping technologies over time. In 1855, soundings, bathymetric soundings, were taken by lowering a line over the side with a piece of lead, taking the time to calculating the time it took to get it back, or the number of knots in that line. You get a map of a bathymetric map. Sometime, as recently as 25 years ago or so, you would get a map like this where the bathymetry, the actual depths, are starting to come into focus. But the sediment types were done by these dots. You see these individual dots are sediment grabs, just a grab going over the side, grabbing some sediment, bringing it back up. What they'd have to do to make a map would be to interpolate from a relatively small number of dots. Thus, you get habitats that are being described here where you obviously see no black dots. There was no sampling done there. But where we come to now, something like this. This is a subset of that larger map that I just showed you, and this is the next iteration of maps that we're going to have. This is a very fine resolution map of sediment type by grain size, which will be ultimately invaluable in mapping the distribution of invertebrates like sponges and anemones and all the things that live in the sediments. Okay, next shot. When you do a multi-beam map, when you use a multi-beam system, it provides two things. It provides bathymetry, which you see here, and it also provides, through acoustic backscatter, a sense of the roughness of the sediment. So what you see here in white are the hardest returns. Those are probably hard bottom areas, gravel, piled boulders. The grayer areas are, I also know this from just being out there, these gray areas are sand, and then the dark black are the deep mud basins, things where the sound is absorbed before it returns. Combining this acoustic backscatter map with the underlying bathymetric map, you get this map that we've already seen that includes this sun-illuminated topographic map with surface roughness. This map, now I'm not sure how impressive it looks from your vantage point, but there is the percentage, if you calculate the percentage of the total ocean bottom that is mapped to this extent, it would be minuscule, or particularly that which we can gain access to. The Navy's probably got more of it mapped than we know. Nonetheless, this underlies basically every project that we do. When we're going to go study fish, site selection for an experimental design, we use this map, and I've used this map repeatedly over the last 5 years. An example is the Seafloor Habitat Recovery Monitoring Project. This particular image was produced for us by some of the folks we have here in the room, Technology Planning and Management Corporation in Scituate. What we've done here, for instance, is use the map to identify this gray or this dark area with the diagonal lines is the Western Gulf of Maine closed area. It's a fisheries closed area imposed by the New England Fisheries Management Council in 1998. It was not part of the sanctuary management, but it happened to overlap 132 square nautical miles of area in the sanctuary. It excluded mobile fishing gear, anything that could catch cod, in fact, commercially. That gives us an excellent opportunity to start looking at the impact of fishing to seafloor habitat. So what we did using the map was go out and identify sites inside and outside of the closed area at similar depths for the 4 major habitat types that occur in the Gulf of Maine. Those are piled boulder, mud, sand, and gravel. So we were able to identify these habitats in conjunction with the fishing community using the map, identify these habitats, and since then, since '98, have been going out annually, or at least annually, sampling the fish that occur there, the seafloor microhabitats, the infaunal organisms, things that live in soft sediments like clams and anemones and burrowing animals, and things that live, epifaunal organisms, invertebrates that live on hard-bottom substrates, again like anemones, tall sponges, corals, those types of things. Next shot. So what do I mean by microhabitats? Well, here's where we start to, to get some images we can start to talk about. Microhabitats, to me, habitat is a variable term. You can line up 10 biologists and you're going to get 10 different definitions of habitat. When I refer to microhabitats, I'm talking specifically about the scale at which individual fish relate to the bottom. And I'm talking about seafloor habitats too. I'm not talking about the water column per se. But you have a number of different things, like these piled boulder habitats. They weren't that visible at the scale at which we were viewing that map, but there are huge numbers of these boulder piles, these large linear features they're raft deposits. They were dropped as the glacier receded 10,000 years ago. It was just dropping these chunks of boulders. You have these huge piled boulder features. I'm going to talk a little bit more about them in a second. But, for instance, you get interstitial spaces in between these piled boulders that animals use as cover, refugia from predation and from currents. These are sebastes, or redfish, that we know use these things. You have even something like sand. Your average nor'easter reformulates the sand regularly, and we get a hurricane in the summer, also reformulates. Some tidal flux occurs. But you get these sand ripples or sand waves that are also used by animals. The mud. Mud is relatively featureless, left unto itself, but organisms that burrow, like here you see a Europhyces, a hake, making use of a burrow that was left by some other creature. And then there's gravel. Gravel will be the source of a great deal it provides structure for a lot of invertebrate growth. It also provides, here you see a longhorn sculpin. This is one of the dominant teleos predators out there. In '98, we did 17 ROV dives in the month of July, and we counted more. These guys, longhorn sculpin, were the most abundant on all 17 dives. They're ambush predators. They hang out in the rock. They use crypsis. They camouflage themselves, hang out there on the rock, and nab little fish as they're swimming by. Nab little cod and little haddock and little pollock. Next slide. When we talk about microhabitats, particularly seafloor habitats, we're talking about the sediment type that we just described, and then any emergent structure, biogenic structure that exists on top of it. So here you see boulders with polymastias sponge. Well, that's a yellow ball sponge. I'm not sure what species that is. Large anemones. In the sand, you get amphipod tubes. These amphipod crustaceans burrow in and then form these little tubes. And we're talking about, an inch at a max, but to a 4-spot flounder like you see right here, that's a viable source of habitat. It provides protection. Whereas here you have shell and shell fragments. The shells are pulled up from the sand. These are clam shells pulled from the sediment by echinoderms, starfish. Some of these starfish can reach 22 centimeters into the sediment to recover these clams. But their shells then provide structure. We know silver hake, for instance, or whiting, a fish that's used for a number of different purposes. Fished highly off the East Coast. When they're juvenile stage, they use these structures. Or mud-draped gravel. Here you have a little bit of mud overlying some gravel that we saw in the last slide. These are Iaphon sponge. We see these fields all over the place. They're huge fields, even times denser than this, and these sponges can grow to 14 centimeters in height. Not necessarily this particular species, but sponges in general provide a great deal of structure for juvenile cod, for instance. Okay. So We're out on our boats. We've got our map going, and now we're going to conduct some science. There are a whole suite of opportunities and things that we use in New England and around the world to do this. Probably the one that most everybody's familiar with is diving. Scuba diving up here. This is from one of the Aquarius missions. That's me counting fish. Scuba is probably the dominant tool used by marine biologists around the world. It has its limitations, but fundamentally it puts you in the water, which is the place as a biologist that you want to be for the most part. There are other options like tethered diver approaches or hookah rigs, where, like, for instance, the habitat, there's a system that you can use that's tethered. You don't need to deal with 100-pound tanks. You just swim out with a mask, maybe you have a weight belt on, and you can walk along the bottom. You're tethered back to a surface. Your air supply is basically infinite. Then you have something like the gym suit. Now, this is clearly not used very often. It's a one-atmosphere suit. It's like a mini submarine. It maintains surface pressure wherever you are. This is probably doesn't even belong on the slide, but it looked good in the corner, so I put it here. I had some blue space I needed to fill. Another relatively new option that is increasingly available to the wider public is the rebreather. Rebreathers they recirculate the air instead of extruding the bubbles. They're actually mixing CO2, and as you can see from here, unlike I'm actually probably holding my breath like you're not supposed to do when in scuba, but rebreathers don't extrude bubbles. There are so they offer certain advantages. It's been hypothesized, for instance, that without bubbles you can approach fish much more closely. We did a study like this last year down in the Keys associated with one of the Aquarius missions. Tried to get to the bottom of that, and what we found was somewhat ambiguous, as much science is. But it looks like you could probably approach individual fish a little closer, but once you got on site and remained in a fixed position, the fish acclimated to your presence and it didn't make that big a deal. Another option, of course, this guy's on a rebreather too, is a heated dry suit, which I've not done personally, but I think it could be rather enjoyable up here in January off the coast of New England. You get you get hot water pumped down to your suit, or you just fill it up before you dive. Not done it, like I said, but an interesting opportunity. Now in Stellwagen, the Stellwagen depths range from 60 feet at its shallowest, and this is a very small bit of the area of Stellwagen, 60 feet, down to about 420 feet. So the vast majority, the recreational dive limits are 130 feet. Now there are people that are willing to dive much lower than that, but in general we try to constrain ourselves with scuba to above 130. So we're not doing a lot of diving out on Stellwagen, but one of the projects that we have spent some time on is submerged cultural resources. There are some wrecks in the southern part of the sanctuary that we're able to dive on. This is a side-scan sonar image, and I'll talk a little bit about side-scan in a bit. This turns out, we think, to be the wreck of the Paul Palmer. There was a series of 5-masted schooners built at Bath Iron Works up in Maine in the 19th century, and this was one of the 5-masted types. It's 243 feet in length, and it's covered in fish. It's a remarkable dive. It's basically 65 to 80 feet down, and we dive on this quite often to photo document with digital still camera and video, collecting things. We're not actually taking things off. The sanctuary regulations preclude removal of anything from any submerged cultural resources, but nonetheless we are attempting to document it for posterity. So if you can't dive and you want to get down there, the next best thing to being there in scuba is being there in a sub, in my opinion. Again, and my opinion is my own, not any of those agencies that we saw at the beginning of the talk. And there are a suite of opportunities available to you. This is a subset of the subs that we've used off the coast of New England. The Deep Worker 2000 is probably the most recent addition to the suite of tools. I'll talk about that a little bit more, but it's a one-person submersible. The Delta, which comes from Delta Oceanographics in California and is regularly shipped back and forth between the coasts, holds 2 people, a pilot and a scientist. You can see the scientist positioned down there. Clelia, and yeah, it doesn't look very comfortable, does it? It fosters a great deal of collegiality among pilot and scientists, as you can imagine. Clelia and the JSL, Johnson Sea Link 2, are both operated by Harbor Branch Oceanographic down in Florida. Clelia holds 3 people, 1 pilot and 2 observers. JSL holds 2 crew members and 2 scientists. And then you have NR1, which is actually, this is the first deep submergence vehicle in the Navy powered by nuclear power. It was put in the water in 1969. It has wheels on the bottom that you can stand down and roll along the bottom if you want. It can hover. For a very long time, the Navy wouldn't even tell the researchers. The researchers weren't allowed to publish the depths which they went in the sub because the Navy didn't want people to know. But now, it holds 7 people. 2 of those are officers, 3 of those are crew, so it only holds 2 scientists. But rewind the tape. Nonetheless, so you see, you have a suite from one-man to multi-person submersibles that actually get you down there in the environment. They all have a similar instrumentation. On all of these things, you can put cameras and videos, mount them on the hull. You can put sector scanning sonar devices. You can put basically the same suite of tools you could probably put on most of these. There would be, if we spent more time, we could analyze the functionality. For instance, here you see a rosette sampler that's probably on JSL2 that Deep Worker is never going to do. And there are limitations to what NR1 can do. But from a biologist perspective, they get you in the water. They get you on site looking at the things you're interested in looking at. Next slide. Just to give a little bit more specific example from a sub we've used recently in the sanctuary. In 1999, the Sustainable Seas Expedition started. It's a 5-year program. It's still going on. It's a systematic exploration of all the sites using this relatively new technology from NUTCO, which is in British Columbia, Canada. It's a one-person submersible where you have the scientist is the pilot. You have basically the area equivalent to a commercial class airline seat. You drive it with pedals. Left and right, forward, backward. You can crab back and forth. You do all that with the pedals. With your hands, you control keyboards for the sector scanning sonar. The array of here we have a wide-angle still camera, digital video cameras, paired centimeter lasers. This is all. It's hard to see in this photo, but it's an articulating arm. I've not seen the sub yet outfitted with the arms in this configuration, but you get this like 3-function articulating arm that you're able to reach out, position, and take photos of stationary organisms. This graphic happens to depict that one time when the tracking was actually working, but one of the advantages to this system is that you're not tethered to the surface. Historically, subs, like we're diving bells, you were tethered to the surface. You were stuck in connection to the ship. I used one of these to sit on the bottom at 420 feet for 4 hours, which you might think, well, that sounds exciting. But in fact, it was very exciting. I got to see some things that, it's not worth building up that much. But I probably sat in an area that was, if it had seen sunlight at all, it hadn't been for 11,000 years. That element was kind of cool. And I was also able to observe the fish that I was there to study vividly. Next slide, please. What we did, again, using the map, was identify in this feature, you could start to see some of these linear features. These are these piled boulder features I was talking about. What we did was we dove at 3 different piled boulder features and started to look at the ecology of fishes on boulder reefs, ultimately to compare those to the ecology of fish on coral reefs down in the tropics. These are analogous in the sense that they provide a great deal of structure for invertebrates, and then that draws in fish. And we were looking to see whether or not there was a community shift from day to night, as you see on coral reefs down in the Keys. And this is part of an ongoing project, but this is an example where the Deep Worker worked out pretty well. When you use the delta, can you go back one, Ken? Actually, back 2. When you use the delta, there's an advantage here. The pilot's basically flying the vehicle. When you're doing transects, he or she is maintaining the distance off the bottom, reporting to the surface for life control reports, which you have to do every half an hour, sometimes every 20 minutes, depending on where you are. As a scientist, you're just comfortably sitting down here between his or her legs, actually collecting data. That is one drawback for Deep Worker. It's very difficult, for instance, to conduct transects because you're working the pedals to try to stay up the right height. You're situating yourself to collect your science, and just when you've got everything right, they call down for life support. You have to sit down, report in all the life support. It's a challenge. But what we ended up doing was using the Deep Worker like a blind, like ornithologists use on land. We would park the sub next to one of these piled boulder features and then turn off the lights. And for 5 minutes, we'd sit there and twiddle our thumbs, give in give a life support reading. And then we'd flash the lights on and do an instant count of what fish we saw, their position relative to the bottom, and the behaviors that they were evidencing. My wife thinks this is kind of cheesy to flash on the fish, and then we're not getting actually a reasonable behavioral assessment of what's going on. But in fact, one of the things that's so often the case in science, it's whether or not it was evidence of accurate absolute behavior in the field. We were doing it systematically, so we were able to compare those behaviors across different sites. Okay, let's bump ahead a few. Another option, I'm going to move it along here, is the remotely operated vehicle. Subs are expensive, as you can imagine, as are research vessels. Some research vessels probably range from $1,800 a day, which is relatively small, to $25,000 a day. If you add in a sub or some ROVs, you add another $8,000 a day. So you're talking more than $30,000 a day to go out there and collect these data. Oftentimes, half of which are probably weathered out, and you're still paying for them. So another very tractable option to get down underwater for long periods of time without actually being there is the ROV, the remotely operated vehicle. Probably, I think the movie Titanic introduced these things to a great deal of the general population when they explored it. And actually, it was some of my colleagues from the National Undersea Research Center that were with Bob Ballard back when they were looking for the Titanic in the late 80s. The Undersea Research Center operates the 4 ROVs that you see here. And they range in size from the Mini Rover, which is probably about this wide. The Phantom, which is a little bigger when it's in its frame. It's probably about here. The Phantom 3S2 is bigger. It's 5 feet across. And then you have the Kraken that's about the size of 2 refrigerators lying on its side. There are bigger ROVs in California that are probably 4 times the size of Kraken, operated by the Monterey Bay Aquarium. But they're tethered to the surface. And so, and piloted or flown from above by a pilot. And you can go on to the next slide. As an example, this is the Kraken, it's a Max Rove that we've named the Kraken. And this is 2 years ago before it turned blue. You can see this is the stern of the RV Connecticut that we saw earlier. The lab associated with these, and this is often the case, is situated in a portable van. All the electronics, where we're sitting in this slide here, it's inside this van. And you can just move it from ship to ship, regardless of where you're going. And what we do is, this is Craig, the pilot. This is Peter. He's keeping location, tracking the position of the ROV relative to the boat, and the position of the boat relative to the area you're interested in studying. Then me as a scientist, I'm sitting over here with my log, and there's an assistant sitting right off camera here, and we're switching back and forth between cameras. You can, just like a sub, you can mount an enormous number of interesting tools in the front of these ROVs. This one's configured with oblique and forward-looking still cameras. You have a suction sampler on the manipulator arm, these high-resolution lights. That is a sector scanning sonar. You have a pan and tilt digital camera. This year we're bringing up a high-definition television camera on the ROV and should really be able to come back with some really exciting images of Stellwagen, which is that goal we were talking about at the beginning of the talk. Just to give you, so when I say Stellwagen, an image pops to mind of this grand vista of seafloor habitat analogous to Wawona Half Dome. So this is one of the things. This was from a cruise last year. This is actually a shot just up close of the manipulator arm with the suction sampler. Next, please. The ROV, that particular, the Kraken, has been used in a number of studies by myself and my colleagues at the Undersea Research Center for studying fish and seafloor habitat connections. Here's a photo I mentioned earlier of a juvenile silver hake sitting on the downcurrent side of a sand wave, making use of this as refugia, probably from currents in this case, but also probably from predation. Here's a haddock hanging out next to a partially submerged boulder. And this is what, in science, we call the thigmotactic response. Animals in nature like to find structure. They like to cover as much of their body as they possibly can when they're resting with structure. And I've seen fish where about an inch of their tail is against a rock, and I think they're operating on an assumption that they're covered. But nonetheless, this guy is hanging out for any number of reasons, but making use of the structure. And down in this shot, this is one of these sponge forests that I was talking about with these 14-centimeter sponges. Here's one of the sculpin hanging out, waiting for some juvenile fish to swim by. This comes from an area north of Stellwagen from a cruise that Peter Oster and from UConn and Les Watling from the University of Maine conducted this summer, looking at the historical range of deep water corals in the Gulf of Maine. Starting to find corals all over the world that are thousands of years old, longer lived than most terrestrial trees. Huge branching corals, things like this that are this tall, and that take 1,000 years to grow to that point. This is a spot from like 400 feet in the northern Gulf of Maine. This is the type of thing that probably occurred all over the Gulf of Maine prior to the large amount of fishing that's currently being conducted there. Okay, next. Another option, and I'm kind of progressing forward both in simplicity of the operation and in cost. The subs are probably the most expensive, habitat's most expensive, then come subs, then ROVs, and now we move to another opportunity, which are these video bottom cameras. These are drift cameras. They're passive. They are tethered to the surface, but you don't fly them like an ROV. They just hang over the side, and you put the ship in neutral, and you drift with the currents. So you can't, if you see an interesting fish or see something interesting, chase after it or go back to it even. You can't even stop. But they're inexpensive. The personnel needs are minimal. This is our system, the integrated seafloor imaging system, and this is the USGS's system. This one, they're similar in the sense that they have down-looking video, forward-looking video, down-looking still photo, and paired 20-centimeter lasers forward and down, so we can use that in the image to size things. The CBOS also has a grab sampler in the middle, so after a transect conducting some video and taking stills, it can actually take a sediment sample as well. And this is the system that was used to ground truth the Multibeam map that we saw earlier. Hundreds and hundreds of dives used by CBOS to collect data to ground truth the Multibeam image. These require, they're very appropriate for certain applications. And an example of that quickly is something that we've done out in another fisheries closed area out on George's Bank. What we did was we used CBOS to drift over some sites inside and outside this closed area at 32 paired stations. We did 20-minute transects. We took continuous video over those 20 minutes, and we took 1 photo per minute. We compared sites inside and outside of the closed area, so the periphery around here is being actively fished, to an area inside here that isn't being fished. Interestingly enough, we didn't know this before going out, but there was a break at 60 meters. This is a 60-meter contour line. Shallow of this these are mobile sands that are subject to regular tidal fluctuation and storm activity. Down deeper than 60 meters, out here at least, was what we've characterized as an immobile sand. The storm activity doesn't get it. The tidal currents don't get it. It's much more immobile. Next. The way we analyze things, this is from the ROV or one of the transects taken by one of the drift cameras or a sub. One way to do it is do a point count, where you overlay an acetate or transparency with points and count the number of things underneath, and then you sum them up and compare them from one site to another. Next slide. Another way to do it is to take the video and treat the video as a series of non-overlapping video quadrats and do a similar type analysis or point count. Here you see that this is a Super-RD sponge. There's a hake resting in a biogenic depression. You get much more area within a video quadrat, but the resolution is not nearly as good as the 35mm still photo. Okay. An example of the type of data that we produce from this is something like this. Remember we talked about microhabitats. Well, sponge microhabitats, a lot of people assume that the sand, not a lot is happening, that there aren't many interesting things happening in the sand. Hard bottom is what we need to focus on. What we demonstrated from this study, and it's a paper that's in prep at this point, is that in immobile sand, that's true. This area is so often disturbed naturally that it's probably, it's doing its own thing independent of the fishing activity. But down in the immobile sand, sponges were far more abundant inside the closed area that had been closed off for 4 years at that point relative to outside areas. Next slide. And the same with depressions. Depressions are formed by other creatures like skates or flounders. They leave depressions. Sometimes the currents erode it a little further, but animals use these depressions as structure or cover. Again, more abundant inside than out after 4 years of fishing. Another opportunity is side scan sonar. Side scan, unlike multibeam, is normally towed like a fish. We call this a side scan fish from behind the boat. It takes images. It's basically like a fish finder turned on its side, perpendicular to the seafloor. and it collects data, as you see here, as you move along the features. Unlike Multibeam, which actually sends back a depth and a location, with side scan you actually get an image. So we're sitting on board watching the image cruise along the computer screen. Next slide. And an example of the use of side scan is, for instance, something like this. There's going to be a talk in a couple weeks, 2 or 3 weeks, on the steamer Portland that you might have read about was located and confirmed this summer in the sanctuary. This is a steamer sunk with all hands lost in 1898. This is a side scan image collected with our new side scan system, and you can see very nicely how things they were able to tick down this, go through an archaeological review, and identify and confirm that this was in fact the Portland. Next image. We are still using some things that are at the lower end of the technology spectrum. This is a bottom grab. We still use these to collect invertebrate bottom samples. If we don't have an ROV to go down there and grab things, you can deploy one of these over the side. You bring them up, dump it out in a tray, use a sieve, and flush the monitor through. We're still doing this now. Regardless of all these technological advancements, there's still nothing like a bottom grab to count invertebrates. One other tool that we've been using a lot recently, I've been personally involved with, is acoustic telemetry. This telemetry work has been around for probably 30 years, but the technology itself has advanced to such a degree that you can get a receiver like this for a very affordable price. It's single channel, 69 kilohertz. You can envision its range is a sphere with a diameter of about 1,000 meters. And then we buy these little coded acoustic transmitters that every 60 to 180 seconds emit a very specific 4-signal code. And as long as the fish that has been, to which this fish has been tagged, swims into the range of this receiver, you get a record. And what we did in 2001 is deployed one down here in gravel habitat, some of the gravel habitat where it's actually precisely where that photo of the sculpin was sitting in the sponge. And then last year, this past summer, we deployed it at 4 piled boulder features that we saw earlier in the slide. To deploy these things, you have to use a railroad wheel as an anchor, put them down on the bottom. You have to recover them using an acoustic release. The hydrophone's here, and then they're supported off the bottom by a subsurface float. So all told, it's about 9 feet off the bottom. To get them back at the end of there's a lot of things that grow on these, so at the end of your 6-month period, you put a hydrophone over the side, you trigger a signal, which should release this acoustic release and send the whole system back up. It's a moment of incredible prayer. You send the signal over and you wait. One of the paradigms I embraced at a very early in my graduate career was that if you put something over into the water and you get it back, that's a very good day. If you put something over the side, you get it back and you actually have some data, that's a great day. And if you put something back, get data that you can actually use, it's a fantastic day. And it's initially I thought I was very lucky. The first couple cruises I went on worked out very well. But in fact, since then I've learned firsthand that it doesn't always work out that well. But so anytime you get equipment back is very exciting. Next slide. Up here with cod, we opted to attach these externally, bring the fish up by hook and line, bring them up very slowly to reduce thermal and pressure shock, put the tag on them and get them right back in the water. Next slide. We would put them in this device we created, get them all the way back down to the bottom, send a messenger down the line that triggers this release, the door falls open and the swim fish is out. All told, 5 minutes after being dragged from the bottom, the fish is happily back on the bottom with this little pinger producing data for us. Next slide. An example of some of the data from gravel habitat. Cod are considered to be wide-ranging animals. We put these are data from one receiver from a 6-month deployment out on this single gravel habitat area. We had animals sticking around for 120 days, on average 31 days. This is just saying the total number of days they were present. That was not what we expected a priori or before we went out there. Next slide. Piled boulders that was even more distinct. Here you see, these are preliminary data I'm still working on. What we found from those 4 piled boulders was that there are really 2 different types of groups of cod out there. One group of what we called residents that were around virtually for on average 95% of the study they were present. Not continuously, but moving in and out, but they were basically present throughout the study. Whereas transients, these guys, some of them were present for a lot of the study, but they were moving around between boulder piles, which is really interesting, and it syncs up well with some of the genetic literature that's come out on cod recently. Next slide. The one other thing I want to, before I wrap it up, just to bring Aquarius back into the picture, Ken and I were both part of a mission last November where we were actually put in a few hydrophones. Conch Reef is right here. We put in a few hydrophones. We put in 5 hydrophones around Aquarius, tagged some fish, and tracked them for 8 months. Based on those data, we went back again in August and put in 27 receivers all up and down the northern Keys at all of these sites to look at the landscape movement of fish, coral reef fishes relative to the landscape, but also relative to these sanctuary preservation areas that are relatively small, closed areas that the sanctuary has put up. Next slide. Here we did the tag implantation surgically. This is a photo of me anesthetizing a blue parrotfish. There I'm setting up a hogfish for implantation. There we're putting the tag in. This is another shot of the blue parrot. We sew it up using your standard medical suture, put a little triple antibiotic ointment on. Here you see me swimming the fish around, reviving it. Sometimes that took 5 minutes, other times it took 15. And then here you see a happy hogfish. The incision for the tag is right there. And then this is an external tag that so we can spot those fish. Next slide. From the data from last November, we found some really interesting things. Here we have a yellowtail snapper. It's an animal that lives up in the water column, swims around in schools. The expectation was at the scale of one of these receivers, you're not going to see a lot of them. But we, in fact, had animals staying up to 237 days to the experiment, which is really interesting new stuff. Black grouper behaves similarly. We got some animals sticking around a long time, others less so. There are a number of things that we have to do. All this work is in prep. But it turns out that every time you put a tag on an animal, you usually get interesting data. There's just, there's so much to learn that tagging any type of animal, usually as long as you've done it in a reasonable way, produce some interesting data. Next slide. So with that, I hope what I've given you is just a cross section of the types of things that we do and the technology we use to get them. I'd like to conclude with a sunset, as most of us biologists do. And thank you for coming and listening. Does anybody have any questions?
If you have, can you repeat the question that you heard?
Sure.
Yeah, just in reference to when you were saying you were down in the habitat and a storm came through and you were stuck in there all day, and then you came out and you had the school of hog snappers all together and the blue tank. What do you attribute that to anything? What would cause that? Do you have any idea?
The question was, based on the phenomenon we observed in the habitat last year after the storm, what explains the aggregations? The answer is we don't know. I was down there with 3 or 2 of the more well-known tropical fish biologists that I am aware of, and we were all aghast. We had no idea what was going on. It certainly has been, that type of behavior had been noted before. This wasn't the first time that these aggregations were observed, but it was the first time we had observed them up close. I don't honestly know what's going on there, and sampling that type of thing would be very difficult to do. So, tracking down after a storm, trying to find these aggregations would be very difficult. But so I'm not trying to evade, but I actually don't know. Yeah, it'd be a really interesting area of study. I think that's the engaging part of science, is figuring out a way to sell that to a funding agency and coming up with a design. Yes?
I was surprised to hear that there's only one functioning underwater habitat in the world. Because I remember reading and hearing about things like top shelf rigging and sea lab and all that stuff. Are there any plans by any agencies or organizations to build any more?
Are there any plans to build additional habitats? I believe there are. In fact, there's the plan I'm aware of, that I became aware of over the last month, is there might be a plan to actually replace Aquarius. I'm not sure why there aren't more habitats. It is costly. It's certainly not so costly that we can't have more of them. I think it's, people are grappling with it. You require, when Aquarius is in the water, you have about 15 people that are on the clock 24 hours a day, round the clock at mission control. It was one of the most methodically operated groups I've ever been a part of. And that's a major commitment of time. You don't get a lot of people who want to spend that much time observing these things. I don't know. Aquarius was designed for working Catalina. It was actually going to ride out on tracks like a roller coaster down to the site. People were going to do their work, and then it was going to ride back up. It ended up being deployed in the Virgin Islands for a while, then here. Some like Helgeland that I showed earlier, that moved around in the North Sea, the Baltic Sea, then came in. I think there are other. There's Jules Undersea Lodge in the Keys where you can spend a couple nights, but there are other types of habitats. This is the only habitat for research at this point. That's a good question. Again, not trying to be evasive, but I'm not sure. Yes. Does the website have a cruise log? And in fact, it should. You are catching us at an interim point between the update of the old website to the new website. It's always been my plan to have, at the end of each year, a summary of everything that's been done. And some data, some reports, a summary analysis. And if you went to our website, you would find some of those things, but they'd probably be a little dated at this point. I'd ask that you bear with us because that is a goal, and it's something that we have in the works, but it's probably an order of months away from actually being real.
How deep did the NR1 go? How deep can it go?
The NR1, how deep can the NR1 go? The NR1 goes about a little bit below 2,000 feet. I think it's like 2,375 feet. Deep Worker 2000 goes 2,000 feet. That's where it gets its name. Delta is about the same. Clelia only goes 1,000 feet, and then the JSL-2 goes 3,000 feet. Then there's, of course, Alvin that I didn't show up there that's capable of much greater depths. Tony?
As you and your colleagues are learning more and more about fish habitat and fish ecologies, how are the resource agencies integrating that information into decisions?
Good question, Tony. The question was, how are resource agencies integrating the type of research we do into management? And the answer is kind of mixed results, I'd say. Mixed to poor. But that's not the fault of the management community. I think one of the challenges is actually figuring out how to integrate this type of work. These are all relatively small-scale studies. How to integrate these into different management approaches. It's a real challenge. Yes?
I know you're a fish guy. Maybe you can help me out with this.
Sure.
Most people don't think about coral when they think about development in full water.
Yep.
And I've heard a lot of fishermen, full-timers,
Yep.
talk about how in the 30s, 40s, 50s they would bring up a lot of what they call trees.
Yep.
And now they're not seeing them at all for a reason that are obvious. But can you tell us a little bit more about those corals and maybe where we can find out about them?
Yeah, you can. Question was about corals, deep water corals. There are, in fact, a number of different sites. You might want to check the site for the Ocean Exploration Initiative. This is a NOAA site. They actually funded the corals cruise, and so you'll probably see some of the same photos that I showed tonight. The particular study was done to look at to actually locate where some corals are existing, but to look at those habitats and then theorize based on where we're finding them now, where they probably were historically. There are some spots out on the continental shelf slope, out beyond Georges Bank, where apparently they were abundant at one time. And there's an area off of Norway that has been discovered that's literally a forest of these things. So it's to the extent that I'm aware, they're long-lived, like their corals. They're not reef-building corals like you see in the tropics. But so they form tree structures like the fishermen refer to them, and they do form somewhat dense aggregations and are very long-lived, so very susceptible to fishing activity or other bottom types of disturbance. But I would use as a point of departure Ocean Exploration Initiative. You might also check the Undersea Research Center at the University of Connecticut. These, one of the additional challenges. I was talking about subsurface technology. Another obvious challenge, as your question highlights and the other gentleman's question, there's a real burden on all of us to get these information out on the web so people can see it. And it results, it's a lot of human time. You need people. It's hard enough to get people to analyze data after we conduct one of these cruises, but we need to do a better job of getting the data out to the people. Yes?
How long does it take in general for the creatures that are anchored to the bottom to regenerate after they've been completely trawled?
The question was, how long do animals on the bottom take to recover after being trawled? That is a source of a great deal of contention, a great deal of study. I spend a lot of time personally studying that issue, the recovery following disturbance, and it's highly variable by habitat type and highly variable by species. For instance, the sand habitats that we see they're animals that the animals that evolved to live in those conditions are used to a great deal of environmental disturbance, so they probably recover more quickly. That's not to say that they are less susceptible. They're still impacted the same way everything else is, but they recover more quickly. Maybe they have higher settlement rates, they grow faster, these types of things. In New England, on hard bottom, some data from George's Bank suggests that you need 10 years for some sponges to fully recover. And as we know with these corals, you're talking about thousands of years. So it's the full spectrum from the centimeter growth provided by amphipod tubes could recover in a matter of months. The physical structure themselves, if sand waves, could recover in months. But some of the longer-lived taxa, somewhere from a decade to 1,000 years. This is an area of ongoing concern. You also then have to introduce the notion that, and you mentioned completely trawled, some types of gears all types of gears have different impacts on the bottom. Like the acute impact of a single trawl has one type of impact. The cumulative effect of multiple trawls has other impacts. And so you get areas where the distribution of effort is heterogeneous. So you get some areas that are getting one pass of a trawl every year, other places are being hit 10 times a month. So this whole discussion, your question hits at a very active point of research. And with respect to the management question that Tony offered earlier, that's an area that the managers are clearly interested in, and we're trying as rapidly as possible to produce data.
One more question?
Just more of a point of information. Up in Gloucester, the Cape Ann divers have a system that they were trying to work with classrooms and students and groups a few years ago, which really hasn't flown. I don't know why. But they take two divers down. One has a TV camera. The other one has a microphone. And you can go down and talk, charter the boat and go down and actually have a class and answer questions of the divers as they pan around and show you stuff, which is kind of an interesting thing. I don't know how many other people do that.
The question was a comment about interaction from actually underwater for classrooms and things. We've actually we've attempted that from Aquarius. We did a lot of live webcasts. Last year, the first time I was in Aquarius, we had planned a great number of them, but in fact, we entered the water 15 minutes before the planes hit the World Trade Center. So we were down underwater for that first 10 days, and a lot of that interaction was obviously disrupted. Aboard the University of Connecticut vessel, they now have streaming video from the ROV that can come up, be telemetered back to land, and go to classrooms precisely like you're talking about. Anytime you put something in salt water, it doesn't always work that well. But people are moving and making headway in that regard. All right, thanks a lot.